The next speaker is Heng Yu. He is a graduate student from Klaus Schulten's Theoretical and Computational Biophysics group, and in his undergrad he studied biology, and he's currently focusing on membrane-protein interactions. And today he will be talking about membrane sculpting by F-bar domains studied by molecular dynamics simulations. Let's welcome him.
So thank you all for coming. So my name is Han. I'm from Klaus Schulten's lab, and today I'm going to give you the latest result update on membrane sculpting by F-bar domains via molecular dynamics simulations. So during cellular processes, cells are dynamically sculpted into many types of compartments, in often cases with help of membrane peripheral proteins. So for example, in here, the cells are sculpted into Golgi complex, ER, and the cell membrane, and they interchange contents with vesicles in between. And for here, we can see that the vesicles are pinched off from the cell membrane by a protein complex called aosome. And the Golgi complex maintains its shape by another protein complex, including Phil1 and RVS. All these proteins contain a special domain called the bar domains. The bar domain has a special structure with 3 helix bundles and has positive charges aligned at one side of the surface. This has been shown to drive membrane tubule formation in vivo and sculpt low curvature liposomes into tubules in vitro. Compared to N-bar domains, there are 3 types of bar domains called N-bar, F-bar, and I-bar domains. Compared to N-bar domains, F-bar domains are usually longer with elongated question shape. The tubules formed by F-bar domains are usually 3 times larger than those by N-bar domains. And another difference between them is that N-bar domains contains an N-terminal helix which inserts into the membrane, which is absent in F-bar and I-bar domains. So there are 2 types of proposed mechanism on how bar domains generate membrane curvature, namely scaffolding and the membrane insertion mechanism. In scaffolding mechanism, negatively charged membrane lipids are attracted to the positive charges aligned at the concave surface of the membrane, thereby generating membrane curvature. While in membrane insertion mechanism, amphifatty groups are inserted into one leaflet of the membrane by the protein, and thereby generate membrane curvature. For N-bar domains, it has an N-terminal helix which inserts into the membrane, while for F-bar domains, it is suspected that phenylalanine residue 117 inserts its bulky side group into the membrane. So the energy of a single F-bar domain is not enough to produce the tubules and vesicles in cells. To produce large curvature in cells, multiple F-bar domains work together. A high-quality quasi-EME image has shown that F-bar domains form well-ordered spiral lattice on the surface of membrane tubule. Recently, several computational studies has done on the N-bar domains, but still little is known for the F-bar domains. There are several questions remains to be answered. How does the arrangement determine the curvature? How does it dynamically sculpt the membrane? And does it use membrane sculpting or membrane insertion mechanism? So in this study, we try to address these questions with molecular dynamics simulations. So first, we want to see if scaffolding is important for its function. Shown here are 2 sets of simulations starting from the same configuration. On the top is wild-type F-bar domain, and on the bottom is F-bar domain with positive charges along the concave surface abolished. That is, no charge simulation. We can see that the positive membrane curvature is developed within 100 nanoseconds for wild-type F-bar domain, but for no charge simulation, no membrane curvature is developed, and the F-bar domain leave the membrane within 80 nanoseconds. This shows that the positive charges are both important for both binding and bending of the membrane, and scaffolding is important for its function. Another thing we want to see is that if the phenylalanine residue 117 insertion is important for the function. So throughout the simulation, phenylalanine residue 117 is buried within the helix bundle and do not form interaction with the membrane. Therefore, it is not likely that F-bar domain uses as a membrane curvature mechanism. So as I said, F-bar domain works as a team to produce membrane curvature. However, of course, we don't want to stop at a single F-bar domain dynamics, but also want to know how they work together. But membrane curvature occurs in time scale of hundreds of microseconds and in size of 100 nanometers, making it difficult to see in conventional all-atom simulations. So since they work together, the atomic detail of single F-bar domain may not be important. Therefore, we use a method developed in our lab called shape-based coarse graining. In this method, we use beads to represent the shape of the protein, and it allows us to reach for larger timescale and longer size with sacrifice of atomic details. It has been used for several applications, including virus and M-bar domains. So in here, we did a single F-bar domain on top of a lipid patch in both all-atom and shape-based coarse graining. We can see that membrane curvature is developed within 100 nanoseconds for both simulations with similar magnitude. So with this model in hand, the first thing we want to know is that how does the F-bar arrangement determine the curvature? So we simulated different F-bar arrangements. That is, F-bar lattices on top of the lipid patch. Shown here is one set of the analysis. By changing the number of F-bar domains on top of the lipid patch, we can understand how does the density change would affect the membrane curvature generated. We can see that at low density, low curvature occurs. This is understandable because the collective bending force is not strong enough. However, at high density, low curvature occurs. Why is that? The reason can be seen from the dense lattice viewed from the membrane up towards the protein. So we can see that in here is the initial lattice, and in here is the lattice after the simulation. We can see that the concave surface of the F-bar domain is separated from the membrane by the tips of neighboring F-bar domains. This occlusion is a reason why dense lattice is not an efficient membrane bender. We found that at a density of 8 to 13 F-bar domain diameters per 1000 nanometers squares, highest curvature occurs. So we use this method to sample different parameters such as densities, spiraling angles, distance, and conformations between neighboring F-bar domains. We have found an optimal lattice that produces the highest curvature. And this optimal lattice produces a radius curvature that is generally within the range of experimental observations. And the black dot in here shows parameters obtained from the cryo-EM image I showed at the beginning of the talk. We can see that the optimal lattice corresponds very well with the cryo-EM image. And at the same time, by changing the conformation of F-bar domains, cell can attain a wide range of curvatures with different types of tubules. So with this model, can we go to even larger time scale? So we observe the complete tubule formation. The optimal lattice is placed on a 380 nanometer long membrane patch. Periodic boundary conditions is applied in the y direction so that it forms an infinity long membrane patch while leaving some space in the x direction for it to occur. We can see that the membrane curvature occurs within tens of microseconds from the edge of the membrane and propagates to the center of the membrane and eventually form a tubular structure at a time scale of 300 microseconds. So in here we show a dynamic picture of how the membrane curvature occurs. 17 F-bar domains per row and 4 rows of F-bar domains are placed on the membrane patch. So periodic boundary conditions is applied in the y direction so that it can curve in the x direction. This simulation lasts about 300 microseconds, and if we perform it in all atom simulations, it will take about 200 million atoms and would take years to complete even on entire supercomputer. So now we have the tubular structures. We can actually fit the all-atom structures into the density of the tubular structures to have a closer look on the structure of the tubule. So these are the all-atom structures. And these are the interactions between the neighboring F-bar domains. So if we compare this to the initial lattice, we find that the tip-to-tip contact is often lost because the tip-to-tip contact is not strong enough, while side-to-side contact is maintained by the key residues via electrostatic interactions. This consistent with experimental results. If we mutate these residues, the lattice will be gone. So in conclusion, we have shown that the membrane sculpting process can be realized at atomic resolution via scaffolding mechanism. We have obtained an optimal lattice that corresponds very well with the experimental data, and we obtained a dynamic picture of how the membrane curvature occurs. So with this, I would like to thank my supervisor, Professor Klaus Schulten, and all of our group members. And thank you all for your attention.
Thank you. Questions from the audience? Come on, any questions? No? Well, so if it falls completely to a circle, is that stable, or does it keep on falling after that?
Yeah, actually, if we remove the protein after that, the circle will stay because it's already fused.
Okay. Well, thank you.
Thank you.
Another question? Yeah.
What about the insertion mechanism? You mentioned there is an insertion mechanism to form the curvature on the membrane.
So the insertion mechanism is actually used by M-bar domains because it has an N-terminal helix that halfly inserts into the membrane. For the F-bar domain, it's not known. So there are some suggestions saying that phenylalanine residue, it might be the insertion mechanism, but we found that it did not actually insert into the membrane. Thank you.
Another question? No? Well, let's thank the speaker once again.